And you don't need to memorize this tree at all, but I just wanted to kind of put this into a little bit of perspective. So the alveolates, which include the ciliates, and so on, the apicomplexes are right here, the ciliates are down here, dinoflagellates are here, so they're part of this group over here. And you'll notice that their sister group to a group we're not talking about because they're not particularly animal-like. These are the brown algae, and so the idea here is, we'll see in a minute, is that chloroplasts probably originated here, and then photosynthesis was largely secondarily lost in this group. The other groups we're going to look at today are, let's see, what else do we have? What are the diplomonads? Oh, they're down in this group here. You don't have to know this, the larger taxonomy here. We're going to touch upon, oh, there they are. Well, actually, just the whole thing here, we'll call that the diplomonads. And then, so don't worry too much about it. And again, not all of this has been worked out fully either. And then, well, I'm going to end with the choanoflagellates, which are the sister group of sponges and animals. That's a group relationship, obviously, you will need to know. All right, but let's go into the apicomplexa here. So this includes a lot of groups that you're probably not super familiar with because they tend to be almost exclusively parasitic. Includes things like gregarians, coccidians, and so on, various kinds of parasitic groups. Unlike most of the other protozoans we look at, they don't have an obvious means of locomotion. So there are no cilia, no flagella. In some cases, there may be flagella and gametes, but for the most part, they don't have cilia or flagella or pseudopods. And they don't really need them because they're basically internal parasites. What they do have, and what gives them their name, is something called an apical complex. That is illustrated here in this little diagram. The apical complex is actually quite complex, kind of a complex structure found at the anterior end of the cell, and it functions to allow them to attach to the host cell and to enter the host cell. You don't need to know any of the structure here, just basically that it's a unique structure to these guys that mostly is part of their parasitic lifestyle. The one thing that is worth noting about the apical complex is that certain aspects, microstructure aspects of it, actually indicate that it is actually derived from a plastid. So it's actually a plastid-derived structure, and plastids are, the non-photosynthetic version of chloroplasts. And so this again suggests that this group evolved from an ancestor that did have chloroplasts, and that's why when we go look at that phylogenetic tree, so these guys have plastids, and there's some indication that certain structures in ciliates might be highly derived, sort of modified things from plastids as well, suggesting that plastids evolved in the common ancestor of these guys here. These kept them as photosynthetic structures. These use them for other things. All right. So what do they? So what do they do? Well, they're parasites. They're estimated to be about 6,000 named species or so. There are various groups. So gregarians, for example, with which there are a lot of species, are mostly internal parasites, either internal, that is within the tissues themselves, or in the guts of various invertebrates, found in insects. We used to actually look at some that we would dissect an earthworm for in lab and look at the gregarians, but we just don't have time for that. Some others include that infect humans, include toxoplasma, which causes toxoplasmosis, cryptosporidia. These affect a lot of humans and other animals. Generally, they don't cause too many symptoms, or they may be mildly flu-like, but they can be a problem, particularly for people with compromised immune systems due to things like HIV or taking immunosuppressive drugs for various reasons. Cryptosporidia can be a common cause of diarrhea, for example. Toxoplasma has been studied quite a bit recently. And so one species in particular, Toxoplasma gondii, is found primarily in cats. So if you have cats, you probably have been exposed to Toxoplasma gondii. You probably have some in you. They're basically spread via the feces of cats. They're use of mice as an intermediate host. When they infect mice, Toxoplasma will actually alter the brain of mice, so that mice basically lose their sense of fear, particularly fear of cat predators. There's even some indication that they may even be sexually attracted to the odors of cats. And that, of course, helps Toxoplasma get back into their primary host, back to cats. So you alter your intermediate host, your vector, so that your vector finds its way back into the inside of a cat by being friendly with cats and so on. It's estimated that perhaps a third of all humans have been infected with Toxoplasma. In most people, it tends to be symptomless, so you don't actually produce any symptoms. Again, it could be a problem for people with, reduced immune competence. There's also some indication that it might, remember, they alter the brain chemistry of mice, so it might alter humans as well. There's been some suggestion there may be a link with Toxoplasma infections and schizophrenia. That isn't 100% certain, but there's at least some evidence to kind of support it. And who knows, it could explain some other phenomena as well. All right, another important member of the Apicomplexua is the genus Plasmodium. There's a whole bunch of species, about 5 or 6 species infect humans, and then there are a bunch of others, so that may be of conservation importance. So certain species of Plasmodium, for example, infect birds, and when infected birds were introduced into Hawaii, birds that were tolerant of this and generally not affected by it, were introduced into Hawaii. Plasmodium spread from the introduced birds to the native birds, which hadn't been previously exposed to them, and many of them succumbed, and lost a number of species, at least as with avian malaria being one of the causes. And one particular species, Plasmodium falciparum, is generally considered the most deadly to humans. It has the highest mortality rate in humans in particular, but we are infected by several different species. Malaria infects about 220 million people per year worldwide. Currently, it's estimated it causes about 500,000 deaths per year, particularly among children. Sometimes the elderly and other immune-compromised people, again, are more vulnerable, but children in particular. That's actually, the good news is that's down considerably from well over 1 million people per year a decade or two ago. So the rates of infection and the lethality has been dropping. Due to everything from better medical access to people using insect netting in their homes to keep out mosquitoes, which are the primary vectors. Plasmodium is particularly abundant in Africa, but it's found throughout the tropical areas where the mosquitoes that spread it live. But in Africa in particular, that's especially where falciparum is located. It's a continued threat. And it continues to be a threat because there have been various anti-malarial drugs developed over the years. But like many diseases and parasites, they evolve a resistance to the drug. So the evolution of drug resistance is a big problem in treating malaria. And also, since mosquitoes are the primary vectors, the evolution of pesticide resistance in mosquitoes has to reduce the ability to control mosquitoes and therefore also reduce the ability to control malaria. But as I said, we're getting better at that. We're coming up with new drugs and things like that. So the death rate is dropping, even if it's still pretty high. So I want to go over the life cycle here a little bit, just to give you a little bit of a feel for what it's like. So we'll start here with when a mosquito bites a person. So the stage from the mosquito's bite, from the saliva in a mosquito, is called the sporozoite. So when a mosquito bites a person, it injects saliva, which includes anticoagulants, because you don't want your little mouth parts to all get clogged up by when you're drinking the blood, and some things that numb you a little bit to reduce the pain. And so in that saliva then are the sporozoites, which enter into the blood. They move through the bloodstream, and their first stop is the liver. When they make their way to the liver, they invade liver cells, and a single sporozoite that infects the liver cell will basically reproduce asexually in the liver and produces, I forget exactly, about 20,000 cells. So a single sporozoite can multiply within the liver cell, and so until that liver cell basically bursts open and releases releases the next stage, which are merozoites. And like I said, a single sporozoite can produce thousands of merozoites. The merozoites then go and are released back into the bloodstream. And there they infect red blood cells, where they basically can, and so they enter a red blood cell, they multiply within the red blood cell, and then they cause the red blood cell to burst open. And in so doing, they produce two types of cells, either more merozoites, so they can accumulate the population every week or 2 weeks, roughly the life cycle here. And they can also produce gametocytes. The gametocytes don't actually do anything to you. They just hang out in the blood, waiting for you to get bitten by another mosquito. So when a gametocyte gets picked up by a mosquito that's drinking the blood from somebody that's been infected, the gametocytes, as their name suggests, produce gametes, eggs and sperm, that unite to form sporozoites in a mosquito. The sporozoites then travel to the salivary glands of the mosquito, and the whole process begins again. So technically speaking, the mosquito is actually its definitive host, because that's the host in which it carries out sexual reproduction. And so, of course, the harm that comes to humans is primarily actually the merozoite stage, because they can, remember, every time a blood cell gets infected, it's going to produce some gametocytes, but it's also going to produce a bunch of merozoites that can infect more blood cells. So people, basically, if you die from malaria, it's often dying from basically severe anemia. You're basically losing a lot of your red blood cells as they're kind of periodically destroyed. And I can tell you from first-hand experience, I've had malaria, and it really, really sucks. I mean, I had a drug-resistant strain at the time, but that was pretty common at that point. And so I was, literally walking down a short flight of stairs was enough to completely exhaust me. You had to take a rest. That's down the stairs. I didn't eat for like 2 weeks. And back then I weighed like 130 pounds to begin with, so I didn't have a lot of weight to lose. And everything hurt, all your joints and muscles and so on. They're just like, ugh. I basically laid in bed for 2 weeks and hoped I could be unconscious for most of that. It really kind of sucked. And it's tricky too, because while they're kind of sitting in your cells and multiplying, you're not destroying things. So there's a cyclical pattern here. So they kind of, release a bunch of cells, the cells die and you get sick, and then they enter new cells, and while they're sitting in those cells, you're kind of starting to feel a little bit better, and think, oh, I'm past it, and then boom, those cells erupt and you get wiped out again. All right, so that's malaria, that's the apicomplexa. Our next group is a diplomonad, the diplomonads. These are another group of protozoans that are mostly flagellated, that tend to, again, be endosymbionts of various animals. Many of them are actually commensals, that is, they don't necessarily harm their host or benefit their host. They hang out in digestive tracts, feed on bacteria, mucus, things like that. And a lot of them don't seem to be necessarily particularly harmful or beneficial, but some of them are parasitic and cause diseases. There are about 100 species or so. They typically have 8 flagella. Most of them actually have 2 nuclei for some reason. And another interesting feature is they lack mitochondria. At one time, it was thought that maybe these are like a very ancient group of eukaryotes before the origin of mitochondria. But as it turns out, they're not. They're just nested fairly deep within the eukaryotes. And so the lack of mitochondria, it was secondarily derived. If you're just relying on your host for most of your resources anyway, because you don't really need them. And as I said, some species are parasitic, and they can cause diseases in fishes and turkeys and humans. The one I want to mention is Giardia, which is a fairly common intestinal parasite in humans. In fact, there are about 20,000 cases of Giardia in the US every year. They're fairly contagious. They can spread fairly easily through contaminated water. That's a major source of infection. They live in your digestive tract, so they get pooped out. They live not just in humans, but also in animals and livestock and things like that. So cattle may do fine with these guys, but they can often be a source. They can spread them. Since they are living in your digestive tract, they mostly cause digestive disruptions. Your body tries to get rid of that toxin by flushing yourself out, and so you basically are flushing out at both ends. So it usually causes very severe diarrhea, very severe vomiting, intestinal pain, and ultimately all of that diarrhea and vomiting usually leads to the most severe consequences, dehydration. If you're not rehydrating a lot, and I mean, you're basically just leaking continuously. So I haven't had this, so I can't tell you firsthand experience, and I don't really want to try either. It doesn't sound particularly pleasant. Remaining dehydrated. So if there's not a lot you can actually do for Giardia except for keeping people hydrated so that they don't, die from dehydration. That's probably the biggest cause. And they're actually kind of a cute-looking parasite. This is what they look like. This is a stained microscope slide. But you can see the two nuclei. You can see a bunch of the flagella around here, some of which originate and kind of, for some reason, have a rather face-like, look like a little face, basically. So they're at least an easy one to recognize because they're the parasite with the face. All right. Our next phylum is a Radiolaria. These are marine protozoans. You saw their tests in lab. Yes, you did have a microscope slide of tests. I mentioned them in here. Why am I picking this particular group and not some others? Turns out, we'll get to in a second, it turns out they're probably a lot more important in marine environments than we used to think. So radiolarians, which is typical of other rhizarians, other kinds of things like foraminiferans as well, tend to have narrow, stiff pseudopods. So this is all the stuff sticking out of the cell here. Those are pseudopods, but they're not pseudopods. They're sometimes called actinopods instead of pseudopods. They're not like those sort of broad locomotory pseudopods that you find in amoebas. Instead, they're mostly feeding structures. They send these pseudopods out. They're fairly stiff. They have cytoplasmic skeletal elements in there. And then food can stick to them, and then they can kind of draw them back and use them as a way of essentially capturing small food items. And so most radiolarians are basically predators, but some are mixotrophs. That is, they can also have endosymbiotic algae that can photosynthesize in addition to capturing food. The reason I mention them is that there was a paper not too long, a couple of years ago, that suggests that these are actually fairly important as a component of marine plankton, much more so than we thought in the past. For starters, they can get to be pretty big. Remember, these are single-celled organisms, but they can get up to about 2 centimeters. It's almost an inch in diameter for the largest type. And most of them aren't anywhere near that large, but they can get up to be pretty big. It turns out that they are really super abundant in the plankton. But we didn't know this for a long time because we mostly studied plankton by trawling with plankton nets. And these guys almost always get destroyed by plankton nets. They just don't handle it. And so we don't realize that they're there. There's bits of organic debris and all that junk you always see in a microscope and ask me what it is. And nobody knows. It's just some debris. Well, sometimes it's, if you look at marine plankton, it might be bits and pieces of a radiolarian. That is and because they don't survive plankton netting very well, they've been undersampled. We basically have been missing them. And as it turns out, they may be actually more important in terms of numbers of biomass than all other kinds of plankton in that same size range, roughly from 0.2 of a millimeter up to about 20 millimeters, that's 2 centimeters. So that's the same size range as most of your algae, and many small animals are in that same size range as well. And they've estimated that they might make up about 5% of the ocean biomass. That's actually pretty big for this one particular group. The way they figured this out, by the way, is since they can't find them through plankton sampling, is to use underwater cameras in the plankton that are able to focus really closely and just keep taking pictures of what's floating in front of the camera lens, close-up pictures, and sample and sort of visually with the camera sampling, sample them that way. And they were finding tons of these kinds of radiolarians. Okay. All right, and our final group, and the one that's most important for our zoology class, is a phylum Choanoflagellata. It's a pretty small group. There's, I forget exactly how many species. I don't know written down. But they're not all that many. They're fairly small. They can be a little difficult to find. They are usually sessile. They're often stalked, but they have a distinctive shape. And the shape is that they have a ring or collar of microvilli. Microvilli are little finger-like extensions of the cell. They don't have the machinery of like a cilium or anything like that, so they're not mobile. But they're just little extensions. Your digestive tract, the cells lining your intestine, for example, have lots of microvilli to help increase surface area. So they have this ring or collar of microvilli, and emerging from the center of that collar is a single flagellum, and that flagellum undulates and moves water past the microvilli, which can then trap food particles. And so that's basically what they're doing. They're using a flagellum to generate water currents, and the water currents move through past the microvilli collar here and capture food particles. The reason we think, well, there's two reasons now, but the main reason we used to think that these are related to animals is because if you look at sponges have a very similar type of cell called a choanocyte. And so you have essentially the same looking cell. Here is a free-living protist, but they're also one of the cell types found in sponges. And I should point out that they can be solitary. They can form small colonies, so different shaped kinds of colonies as well. They can kind of join together in a gelatinous mass, or have a common stalk here. So they form small colonies, or they can be solitary. So this is a picture. This picture is from a recent, again, a not too old paper that shows that at least in one species of choanoflagellate, the presence of particular bacteria and some of the signaling molecules that bacteria secrete can cause them to switch from being solitary to being colonial. So this gives us a little bit of a hint of how the origin of first colonial behavior and then multicellular life might have taken place in animals. So you have these circular guys here are the solitary individuals, and then here's a small cluster of these choanoflagellates. And that clustering is triggered by secretions from a bacteria. So it's possible that sort of multicellular animal life got its start with the help of bacteria. I think we got, I thought I had another picture. What happened to that? Oh, I think it's on my next group. Okay. Yeah, that's when we get into animal origins. We'll show you more of them. So that's basically our very quick tour of protozoans. So any questions so far? All right. So now we're going to get into the actual animals themselves and talk about the origins of animals over today and Monday. All right, so like I said, animals almost certainly evolved from some sort of a protist ancestor. We'll get into the choanoflagellates in a second again. In terms of when this took place, we don't really know for sure, but 800 million years ago, but I should point out that MA, capital M, lowercase a, is the abbreviation for mega-annum, which is just technical, official units for millions of years. So 800 MA is 800 million years ago. All right, and that date is derived mostly from molecular data. So if you sequence protozoans like choanoflagellates and sponges and other animals, you can look at the number of genetic differences that have accumulated. You look at roughly, you can calibrate the rate at which differences are accumulated using a fossil record for groups we have better records for. And you can use that to get a rough estimate of when those two clades might have diverged. There's a lot of uncertainty here. The rates of molecular evolution aren't always perfectly constant, but it gives you a pretty good idea. And it seems to correspond roughly to some worm tracks that might be of roughly similar age. The trace fossils are fossils of behavior, tracks, ways, footprints, things like that. So there's some some suspected worm tracks that might be close to 800 million years old, but those tracks are highly debatable because there are abiotic things, like you can have a glob of gunk sitting on the ocean, and the currents can roll that glob along the bottom and it will produce a track that looks pretty similar. So some abiotic processes can produce similar tracks, so that's a little much trickier to interpret there. But that maybe give us a rough idea of when animals originated. The first unambiguous animal fossils appeared about 630 million years ago. We'll get back to those later on. There's some possible very small fossils of little irregular shaped blobs that look like they might be sponges that date to about 760 million years ago. So it's possible that sponges were around that long, and if they diverged 800 million years ago, the earliest animals probably would have been pretty sponge-like. So that actually makes reasonable sense. We don't have any sort of direct evidence from the fossil record of exactly how we went from a single-celled protist to multicellular animals. There's kind of a pretty big gap in our knowledge, in our direct knowledge. But we can infer things from how animals develop, and so on. All right, as I already talked about with protozoans, we got this all on the same day here, so kind of repeating it, is that the closest relatives of animals are the phylum Choanoflagellata, the choanoflagellates, and they have a very similar structure. So again, these diagrams show different colonial choanoflagellates. This shows a photo of the solitary choanoflagellate, and the drawing on the right shows a choanocyte. This is a type of cell found in sponges. Sponges have multiple cell types, but one of those is a choanocyte, and it has the same basic structure as choanoflagellates. It has a ring-shaped collar of microvilli and a flagellum that emerges from the center of that. And so that's a fairly specific looking type of cell. It's not just some amoeboid thing, which sponges also have, or your basic, rectangular epithelial cell. It's a pretty distinctive looking cell that looks very distinctive compared to a particular group of protozoans. So that kind of, suggests that, hey, sponges may have come from choanoflagellates. In fact, people have even considered the possibility that maybe choanoflagellates are just highly reduced sponges. Molecular data tends to confirm the relationship, that is, that choanoflagellates are outside of the animals, that is, they're not just reduced sponges, but that choanoflagellates and animals are basically sister groups. We are each other's closest relatives. So this is basically what animals originated from, some little colonial, presumably, colony of choanoflagellate protozoans that became truly multicellular in the sponges. So the next question is, well, how do you go from a colony to a multicellular animal? What is sort of what is a possible sequence here, and so people have put forth a number of hypotheses. And generally the favored one is what's called the colonial, that's kind of been around for a long time, is the colonial theory, which basically says that animals originated from a spherical, probably hollow colony, something similar to like you might have seen in Introvia or something, a bulbox colony. It's basically a cluster of round cells. They're green algae, so they're photosynthetic. They're not close related to animals, but they can form a hollow sphere in a similar way. So you have sort of a hollow spherical colony here, maybe with flagella sticking out for locomotion, and then you get a little more sophisticated. Then you have an invasion. One side flattens, and maybe hanging out at the bottom, and folds inwards to create a cavity where you can digest food, a feeding chamber, basically. The idea here is that if you want to digest food, and you're a colony, you're going to have to be right up against the food, and your enzymes are going to just drift all over the place. But if you can kind of capture your food in a little space here, you can be much more efficient about how you use your enzymes. They're mostly going to be right in that space attacking your food rather than diffusing away. So a feeding chamber would be more efficient to eat, especially if you're feeding on somewhat larger bits of food. And notice that this basically looks a lot like gastrulation that we see in animal development. That is, you start off with a blastula, a hollow sphere, and then you have a movement of cells into the inside here, an invagination, that then forms a gut here. And so that similarity between gastrulation and is what has led to this idea that gastrulation basically may reflect essentially the evolutionary sequence that led to animals in the first place. It's not the only hypothesis. There's a variant of that, which I'm not sure it has a particular name, where, again, you start off with a hollow sphere, but then you have cells moving in from the edges into the inside and basically filling it up to form what's called a gastria, a solid, more or less solid sphere. The reason this, the justification for this hypothesis is that in some cnidarians, gastrulation does look like that. So remember I pointed out that when we talk about gastrulation, there's all sorts of oddball exceptions. In some cnidarians is where we see one of those exceptions, and so that could suggest an alternative route for the origin of animals. The alternative to the sort of, and so both of those are generally considered parts of variants of the colonial theory. You start off with a hollow sphere, and you develop into, you have a gastrulation-like process, and you form a gut, and so on, and that's how you get to animals. And since you're starting with a sphere, which is essentially radially symmetric, the colonial theory tends to suggest that the first type of symmetry we find in animals would have been radial symmetry. The alternative is something called the placula theory, which basically suggests that animals started off as basically a flattened structure, a colony of flattened cells rather than a hollow sphere. And then and if you look at it from the top, it might be fairly irregular, so it might not necessarily have any kind of symmetry. It might be asymmetric. And then those cells separate. They form a layer in between them here, an acellular layer, sort of a mesoglia type layer. And then, again, to feed, you have differentiation of cell types. The bottom cells, for example, might be larger, more focused on feeding. And then you have, again, an infolding here, which creates a digestive chamber. The big difference between this and the standard colonial theory is that the intermediate stage is basically this kind of flattened placula stage, rather than a sphere, and you only get that hollowed outness a little bit later. And so this hypothesis suggests you could actually go to the earliest animals might have been asymmetric rather than having a radial symmetry as you would if you simply went via a hollow sphere. The main evidence for the placular theory comes from the fact that there is an animal that kind of looks like the intermediate stage, and it's a species called Trichoplaxenhirans. It has its own phylum, the phylum Placozoa. It is the only named species within that phylum, although genetic studies suggest there may be one or two others. And again, it is a flattened double layer of cells. It has some gel-like mesenchyme in between them. There are amoeboid cells that can move in between here as well. And they can form, this is exaggerated here, they're not quite that extreme, but they're generally flattened, but they can kind of hunch up and create a feeding chamber where they can digest food. They crawl over some food and hollow out the spot around that food and secrete digestive enzymes. And so this looks an awful, and so this is basically what this hypothesis is suggesting, that you're starting from a trichoplax-like animal as the earliest sort of life forms. If you look at it from the top, they're basically somewhat irregularly shaped, and they can change their shape somewhat, so they could look almost amoeboid-like, although they don't produce the true pseudopods or anything like that, and they are multicellular. And so people have suggested, well, this might be a good model for the origin of the very first animals. And there's some genetic evidence that kind of is consistent with it as well here. And so what you're looking at are the two diagrams on the left are kind of a hypothetical evolutionary scenario. This is a lateral or cross-sectional view, and this is a top-down view, an aboral view or oral view, doesn't really matter. And this is a cross-sectional view, and you're basically evolving up here, so which is not necessarily the direction I would have chosen, but so the ancestor is here, basically two flattened layer of cells. If you look at it from the side in cross-section, if you look at it from the top, it would basically be irregular blob. From there, you go to one that can form a feeding cavity here, and then from that, you get to a more extreme, more gastrula-like looking shape that starts to look a little bit in its general shape like a cnidarian medusa. And then at the top, you have a true medusoid kind of structure. And again, this would look at it from the top. The little orange and red dots here actually represent the theoretical expression of a gene that is basically important in regulating the origin of these cells, and is expressed, or theoretically expressed, at the boundary between the upper cells and the lower cells. And so if you look at it from the side, they'd be expressed here and here. If you look at it from the top, they'd be basically expressed as a ring near the edge. And as it turns out, there's a gene has been identified in Trichoplax called the TROX2 gene, which is found right at the boundary between the upper and lower levels. And so the drawings at the right are sort of, this is a hypothetical evolutionary scenario. The drawings at the right suggest actual modern, sort of modern analogs. So you have two different diagrams of Trichoplax. One in its sort of ordinary locum flat movement, and this is in its sort of bunched up feeding stage here. So that's what this would look like. And then at the end, you have a small cnidarian medusa here that basically resembles this sort of theoretical stage. What we don't have is something that represents a somewhat more primitive stage than a cnidarian medusa. That's a little bit intermediate between, so we don't have a modern analog for that. But anyway, back to the genes. So the TROX2 gene has been identified in the Trichoplax, and it's expressed right around the edges. That is right at the boundary between the upper and lower cells. And it turns out that that gene is very similar to, and probably homologous with, a gene in cnidarians called the CNOX genes, which are also expressed at the boundary between the ectoderm and the endoderm. So basically right in here in actual cnidarians. So it's this is basically consistent. The genetic studies is at least consistent with this idea that the placular theory could work. At least it does seem like you can go from here to here reasonably easily. Okay. All right. So where are we on time? Okay, we got a few more minutes, so we'll start talking about fossils. So basically, so this stuff here is mostly theoretical. I mean, you look at genetics of modern animals, but what we really want to know is also, well, what did the first animals look like? What does a fossil record tell us? So fossils are basically any sort of preserved remains or traces or indications of past life of organisms that have been long dead. And of course, we have the total number of named fossils is somewhere in a few hundred thousands compared to well over 1 million named species of living organisms. But the fossil record, the actual number of extinct animals is much, much, much greater. It's estimated, the usual number is 99.9%. I have never been able to find exactly where that number comes from. It's cited and used a lot. That is that 99% of all species that have existed are now extinct. And why it's 99.9 as opposed to 99.96 or something, I don't know. I'm not sure if this is actually based on anything really quantitative or 99.9 sounds good, somebody said, and everybody else copied it. But anyway, most things that have lived are now extinct. And so that means that our fossil record is really inadequate. If we multiply the number of living species, known living species, let's say roughly 1 million species, named species. I'm not sure exactly where it's at, but it's, I think, more than that. But somewhere around 1 million or something, I think, named species, right? Anybody remember? Covered in another class. Anyway, let's say 1 million named species. If 99.9% of those are extinct, that means that you should have at least 1,000 times as many fossil species, so about 1 billion fossil species, of which we have maybe 300,000 named fossils. So the fossil record is very, very, very inadequate. And it's also very biased. That is, the kinds of organisms that fossilize, not all organisms fossilize equally well. The kinds of environments in which they fossilize varies as well. So organisms with hard parts, with bones and shells, with mineralized tissues are going to be much more likely to fossilize than those that are only soft and squishy. We don't have, we have lots and lots of vertebrate fossils with their bones. We have lots and lots of clam and brachiopod fossils with their hard shells. But we don't have very many jellyfish fossils because jellyfish don't have hard parts, and so it makes it much harder to fossilize. Doesn't mean they can't, but the conditions have to be just exactly right, and those conditions are actually pretty rare. So there's a bias there. We tend to see more animals with hard parts than with soft parts, and most fossilization takes place in an aquatic environment. Organisms need to be buried with the sediments, and what moves sediments the best is water. So almost all not all, but most fossilization takes place involving water. It can be marine water, it can be fresh water. Most land animals that have formed fossils, again, there are exceptions, but the majority of them are fossilized in lakes and streams and things like that. They die near a source of water, but that water then carries the mud that buries them and allows them to be preserved. So again, that tends to bias the fossil record. We're much more likely to find aquatic species. We're much more likely to find shallow coastal water species than deep sea species, because the rocks, if they get buried in the deep sea, those rocks probably never make it up to the surface. They're separate from the continental shelves, so they would get destroyed before they ever actually are somewhere where they can be excavated. So there's all kinds of biases here. Just always keep that in mind when thinking about fossils and interpreting a fossil record. All right. So if we want to use the fossil record, one of the things we need to know is how old things are. And so there are two ways that we can date fossils. One is basically called relative dating. And it doesn't have anything to do with family reunions in Kentucky. But relative dating is where you basically use a stratigraphic position, how high or low the fossils are relative to other fossils. So it allows you to say that this fossil is older than that fossil is younger than this fossil. We can actually compare sites that are far apart by using indicator fossils. So an indicator fossil is a fossil that may be fairly common, found in a narrow time frame. So if that fossil is found in one layer in one location, we can go hundreds or thousands of miles away. If we find that same fossil in another location, we know that layer is pretty much the same age. And so then we can tell that this guy, for example, is younger than this guy, even if they don't necessarily occur in the same location. And so that's relative dating. It allows us to build a sequence of oldest to youngest, but it doesn't allow us to put dates to it. To put dates to things, we have to use absolute dating, and that basically means that it's putting actual numbers to it. And primarily we use radiometric dating. That is, we use a radioactive decay of elements that were in those rocks when those rocks first formed by cooling off from a molten state. So when lava or magma cools off, it basically locks in those elements, those radioactive isotopes, and the radioactive isotopes continue to decay at a known rate based on a half-life. So for example, one common type of radioactive dating is potassium argon. And it has a half-life, do I have a half-life of potassium? 1.3 billion years. So if you start off with a bunch of potassium, after 1.3 billion years, about half of it will have decayed into argon. Now, potassium is a common component of many minerals. Argon is a noble gas. So, but while the rock is molten, any argon would simply escape. But once that rock solidifies, you're left with, basically you have a bunch of potassium in that rock, and some of that potassium decays into argon. And so you can look at relative amount of potassium to argon in those rocks, and if you know what the half-life is, you just do some basic math. It's a little more complicated than that. For one thing, measuring the amounts of these things is going to require some high tech. But basically, you do that, and you can determine the age of the rock since the rock formed. That's true for rocks of volcanic origin. There are some cases where a few limited situations where bone and other hard parts have been aged directly based on radioactive isotopes that were taken up by them. That's fairly limited and probably doesn't work. That doesn't only go back probably maybe 100 million years at the most, if that not far. The one limitation of absolute dating is that most fossils are not found in volcanic rocks. Most fossils are found in sedimentary rocks, which are sediments, sand and silt and mud and clay and so on. And sands and so on are basically tiny little pieces of rock that was formed much earlier and eroded away and then basically clumped back together to form sedimentary rock. So if you use radioactive isotope dating on most, not all, but most sedimentary rock, you just determine how old the original parent material was, which could be hundreds of millions or even billions of years older than when the sediment formed and trapped that fossil. So that puts a limitation on things. You have to basically find the volcanic layers and age and date those, and your sedimentary rock and the sedimentary rocks between those you can tell that it fell within some range. But again, if you kind of combine this with using indicator fossils, you can line sequences throughout the world. In some sequences, you can have a close layer that's a volcanic in origin, and you can use that to date those particular layers and put upper and lower limits on it. So one of the things you might see is if you look at a sequence like this of the history of Earth, is that sometimes these numbers are different in different tables, and that's because depending on how old it is, we're constantly essentially refining those dates, since we're not usually dating a lot of these layers directly. I'm going to stop here since we're out of time, and we're going to head upstairs. So next time we'll talk a little bit more about the geological timescale, and actually mostly focus on the events leading to the fossil record. What the fossil record tells us about the origins of animals.